Greeting everyone. Welcome to our presentation. Today we are going to present for our work that we have been doing, and it's called genetic CRISPR Cas9. So if we misspelled everyone. So if we misspelled a word in a transaction, we can change it and we can correct it or even delete the word. And this function within our DNA can be overridden, and it can be possible, and it is taken by a system called CRISPR-Cas9. But what is CRISPR-Cas9? It's a clustered, regularly interspaced, short palindromic repeats. It's a system consists of two things: Cas9 that cuts the DNA, and guide RNA that recognize the sequence of DNA to be edited. And each cell in our body has a copy of genome that can be individual in appearance, so it can cut the DNA and re-edit it, so it can be produced, and the editing of the genome, so with the diseases, and researchers have examined thousands of genes that increase the risk of disease to be recognized and be replaced with other genetics and help to improve them. And this new immune system discovered in bacteria that allows two things: prevent foreign DNA from entering your genome, and the other one is it's also target the invading DNAs and destroys them. Now this immune system helps that system has been adapted to the DNA, telling that it can be edited and changed with the other RNAs, and so with the diseases that has been come, and it can be a type of enzyme that can cut the DNA. And as you can see in the video, the guide RNA is attached to the DNA cutting enzyme. Cas9, and this complex is introduced to the target cells. It locates the target letter sequence and cuts the DNA. At that point, we can edit the existing genome by modifying or deleting or inserting a new sequence. In brief, act as a cut and paste tool for DNA editing. One of the RNAs containing is a sequence that matches the invading of virus and is known as a guide RNA. So it can find a target in the viral genome, Cas9, that cuts the target DNA and inactivates the viruses. And in other words, they create a small piece of RNA, so it can be with a short guide and a sequence that binds the specific target sequence in the cell DNA, and this binds of the Cas9 can be when the guide RNA enters the cell, it recognizes the desired DNA sequence. After that, in this way, bacteria use a CRISPR as an immune defense when bacteria are infected with the viruses. So it can be changed with the other enzymes and cutting them, and for the viruses in the specific pattern into their own DNA to create segments called CRISPR's arrays. As you can see in the video, CRISPR is the combination of scissor-like proteins, and such as one called Cas9 and guide molecule. The molecule acts as a bloodhound, taking CRISPR on the hunt to specific sites within genome. Once the protein cuts the cell's DNA, and the spot that RNA has been changed with the new RNA that has been modified to them, or it has been told to them. And For the further information, my friend Amirsaleh will tell you about it.
Well, as my teammates told, CRISPR-Cas9 can edit the gene. So for knowing that, let's take a look at the Duchenne muscular dystrophy. Since it is a genetic disease, it is considered a suitable candidate for editing with CRISPR. Yes, the simple muscular atrophy (SMA), the neurodegenerative disorder by characterized. This is caused by mutation in the SMN1 gene, which is responsible for making a protein called SMN and necessary for the survival of motor neurons. In healthy people, the SMN1 gene encodes a protein called motor neuron cell, which, as its name suggests, plays a vital role in the survival of motor nerve cell. Thanks for your listening. You can connect us, and I want to great thanks for Ms. Mogaddam for her support, and also Ms. Hosseini for her help.